Okay, let's unpack this. When you say the name Francis Crick, I mean, almost everyone's mind just goes straight to one thing,
It's
right?
a double helix, absolutely.
Yeah, the double helix. But the stack of sources we have here today, it shows that defining Crick by just DNA is like, well, it's like trying to define the whole universe by looking at just one star.
That's a great way to put it. Our mission today is to really dive into Crick's entire intellectual journey. Because it was staggering. We're talking about a guy who goes from designing, sophisticated military minds in wartime
Right.
to then drawing up the definitive architectural blueprint for life itself. And then for his final act, he pivots again to chase down the physical basis of the conscious mind. He's, arguably one of the most important theoretical biologists of the whole century.
It's that dual quest that makes him so compelling. The sources make it so clear. He was driven by these two just immense fundamental problems. First, how do simple molecules, just stuff, become living, information-carrying things? And second, how does that living stuff, specifically our brains, how does it generate a conscious mind?
And we're going to trace that path because he didn't just contribute to these fields. In many ways, he created them and then redefined them.
And that path was anything but a straight line. I mean, he was born in 1916, and his first move was into classical physics.
Right. He started a PhD at University College London, and he later joked that he was working on the dullest problem imaginable,
Which
measuring the viscosity of water at high temperatures.
How thick hot water is.
Exactly. Think about that. One of the greatest minds of the century was initially obsessed with how thick water is when it's hot.
But that's our initial hook, isn't it? This early focus on just hard physics, precise measurement, and then this almost cinematic moment when World War II just blows it all up.
Literally. It's the ultimate interruption. A German bomb fell through the roof of his laboratory during the Battle of Britain.
No.
It destroyed all his experimental apparatus, the glassware, the measurement equipment, everything, just gone. His physics career, at least that part of it, was obliterated.
And before he could even think about what came next, he had to serve. The sources really highlight this part of his life. He was a very capable military scientist first.
Oh, absolutely. He spent the war working for the Admiralty Research Laboratory. He was focused on designing magnetic and acoustic mines, highly technical defensive and offensive stuff. He was even credited with designing a new, more effective mine against German minesweepers.
So this wasn't just some minor desk job. This was critical, applied physics under the most extreme pressure.
Exactly. So by the time the war ended, he already had this track record of solving really high-stakes, difficult problems.
Okay, so let's delve into his formative years a little, because that really sets the stage for what you just described, this scientific aggression, you could call it, that willingness to just jump into a huge, messy problem and assume he could solve it. He grew up in Northamptonshire, right? His family had a boot and shoe factory.
A very grounded, very practical environment. But the scientific seeds, they were definitely there, latent in the family history.
Oh, so.
His grandfather, Walter Drawbridge Crick, was a successful businessman, yes, but he was also a really avid amateur naturalist. He actually corresponded with Charles Darwin.
With Darwin himself?
Yep. There are even a couple of gastropod species named after him, which shows you he was taken seriously in those circles. So that curiosity about the natural world, it was sort of in the family DNA, so to speak.
And his famous skepticism, his atheism, that was formed incredibly early, too. The sources say he rejected religion by the time he was 12.
12 years old, he decided he preferred the scientific search for answers over any kind of religious belief. And that intellectual independence, that reliance on logic and observation above all else, that really stuck with him for his entire life. It's a crucial piece of the puzzle.
So after the war, after the mines, the actual migration into biology happens. It's 1947. He's 31 years old.
Right. And he wasn't alone in this. It was part of this major post-war wave of physical scientists moving into biology. A lot of them felt that physics had sort of peaked. They'd figured out the atom.
Right, so where's the next frontier?
Exactly. And biology looked like it was just ripe for discovery. It was messy, it was complex, and it was waiting for some fundamental laws to be found.
He was inspired by figures like Sir John Randall, right? The radar guy.
Yep. These were minds that were used to solving massive system-level technical problems, and they were looking for a new challenge. For Crick, though, this shift was profound, almost philosophical.
What do you mean?
He said moving from the neat, predictable world of physics, which he described as having elegance and deep simplicity, to the messy, elaborate chemical mechanisms of biology. He said it felt like being born again. It was a complete reinvention.
That sounds exhilarating, but I have to wonder about the confidence or maybe arrogance that comes with that. An outsider walking into a field and thinking he knows better, did that alienate people?
That's a key question. And the takeaway here is this intellectual attitude he brought with him from physics, which he openly called hubris. He wasn't afraid of that word.
So he embraced it.
He did. He believed that the wild success of physics proved that equally huge, sweeping advances had to be possible in biology, too. Even though the problems were, way more daunting, a living cell is just staggeringly more complex than a hydrogen atom.
So the hubris wasn't about him personally. It was about the method, the physicist's approach.
That's it, exactly. And that belief encouraged him to be far more daring, far more theoretical than the typical biologists at the time, who were often, more focused on just describing and cataloging things. Crick was looking for the universal rules, the architecture.
And that approach gets him a spot at the Cavendish Laboratory. He joins Max Perutz and John Kendrew's group, supervised by a Nobel laureate, Sir Lawrence Bragg.
And it's so important to remember the context here. Because of the war, he's still technically a graduate student. He's 31 years old.
An older, highly intellectual grad student surrounded by the biggest brains in the country.
And he dives right in. He teaches himself the incredibly complex mathematical theory of X-ray diffraction. This was so crucial because it's what allowed him to actually see the patterns, to interpret those cloudy photographs that the crystallographers were producing.
So even before he saw a picture of DNA, he was building the mathematical tools he'd need to understand it.
100%. His early theoretical work with William Cochrane and Vladimir Van was foundational. They developed the math to describe the diffraction patterns you'd get from helical molecules.
From a helix.
From a helix. And their theories successfully matched the alpha helix structure in proteins that Linus Pauling had just discovered. So this proved Crick's theoretical approach was sound, and that math he developed would be indispensable the moment he turned his gaze to DNA. He was ready.
Okay, so this brings us to the collaboration everyone knows. It's late 1951. Crick, who is now 35 and still a grad student, starts working with James Watson.
A young American whiz kid. He was only 23, but he already had his PhD.
And they just clicked. They shared this intense, almost obsessive focus on one single question. How is genetic information stored, and how is it copied?
They formed this legendary partnership. It was built on these really complementary skills. Watson was the impatient, restless one, always pushing, always asking, why not now?
And Crick?
Crick was the precise theoretician. He demanded absolute logical rigor for every single step. They were basically in a nonstop brainstorming session, and apparently they talked so loudly and constantly, their colleagues were always telling them to quiet down.
And looming over all of this was the massive shadow of Linus Pauling.
Pauling was the giant, the titan of the field. He had just cracked the alpha helix, and Watson and Crick were absolutely terrified that he was about to solve DNA too.
He was scheduled to come to England, wasn't he?
He was, and this is where a remarkable bit of accidental luck comes in. Because of some of his political activities, the U.S. government restricted his travel. They wouldn't give him a passport.
You're kidding.
Nope. That decision kept him physically away from the labs in London, away from the crucial data that was being collected at that exact moment.
History turning on a bureaucratic decision.
Yeah.
Amazing. But even with Pauling sidelined for a bit, Watson and Crick didn't get it right away. Their first model in 1951 was a complete disaster.
A chemical disaster. Profoundly erroneous, as they said. They built a triple helix, not a double helix.
A triple helix.
And they put the phosphate backbones, the structural supports, on the inside.
Which is where Rosalind Franklin comes in. She saw that model and just shot it down immediately.
Instantly. She pointed out the basic chemical flaw.
Can you break that down for us? Why was that such a basic flaw?
Well, the phosphate groups are highly negatively charged, and they're hydrophilic. They love water.
Okay.
And DNA exists in the watery, salty environment of a cell nucleus. So just simple chemical necessity says those charged groups have to be on the outside of the helix, facing the water. The hydrophobic bases get tucked away safely in the core.
And Franklin understood that chemistry perfectly.
Perfectly. She shut down their first attempt in about 2 minutes.
So this brings us right to the heart of the enduring controversy. The crucial data from Rosalind Franklin and Maurice Wilkins at King's College, which Watson and Crick used to build their final model, but without her knowledge or her permission.
This is a really layered story. We can basically identify 3 specific moments where they got access to information that they really shouldn't have had.
Okay, moment number 1.
Watson attends a seminar Franklin gives in 1951, and she presents key details from her X-ray work. She talks about the 2 forms of DNA, the A and B forms, and crucially, she states that the phosphate units are on the outside.
Which directly corrects their first big mistake.
Directly. Okay, moment number 2. Wilkins, who was Franklin's colleague at King's, they had a very strained relationship. He shows Watson photo 51.
The famous one.
The iconic one. It's this beautiful X-ray diffraction image of the B form of DNA taken by Franklin and her student. It was the clearest evidence anyone had ever seen of a precise helical structure, and Watson saw it and immediately knew the helix was real. It was the smoking gun.
And the third avenue of information, which is maybe the most revealing about Crick's role as the theorist.
Yes. Crick's own PhD advisor, Max Perutz, gives Crick a copy of an internal progress report from the Medical Research Council.
An internal report.
It was not meant for public consumption. It was meant to coordinate work between labs. And that report contained Franklin's detailed crystallographic calculations and her structural analysis.
And what was the key insight in that report?
It was her finding that the backbones in the air form of DNA were anti-parallel.
Let's pause on that because that's a deep dive nugget right there. What does anti-parallel mean, and why was it so critical?
It means that the 2 sugar phosphate backbones, as they wind around each other, they run in opposite directions. Think of a highway with traffic going north in one lane and south in the other. It might seem like a small detail, but it changes the entire geometry of the molecule. The hydrogen bonds between the bases are extremely sensitive to that geometry. If the chains ran parallel, the bases wouldn't pair up right. Franklin's systematic measurements confirmed this anti-parallel structure.
So getting that report gave Crick the absolute confidence to build their final model with anti-parallel chains.
Exactly. He said later that seeing those calculations was the moment he knew for sure it was a double helix with anti-parallel chains. So now they have the geometry, but they still need to figure out how the bases connect in the middle, which pairs with which.
And this is where the final pieces of the puzzle just fall into place almost all at once. It's a great example of how science is about synthesizing different bits of data.
Absolutely. So piece number 1 is Chargaff's rules.
Right.
The biochemist Erwin Chargaff had shown that in DNA from any species, the amount of adenine, or A, always equals the amount of thymine, T, and the amount of guanine, G, always equals cytosine, C.
So A always pairs with T, and G always pairs with C. It points directly to specific pairing.
It does, but that wasn't quite enough, because they had the wrong idea about the chemical structure of the bases themselves.
And that's piece number 2, another small technical detail that could have derailed the whole thing.
It's about something called tautomers. It's about the precise chemical form of the bases, which determines where the hydrogen bonds can actually form. They were stuck, so they went to ask a physical chemist who was sharing an office with them, Jerry Donahue.
And what did Donahue tell them?
He told them they were using the wrong forms. He confirmed that the predominant forms were the keto and venumine configurations. If they had used the incorrect forms, the hydrogen bonds just wouldn't have lined up properly.
So the simple correction of basic chemistry was the final key.
It was. And you get this great image from the sources of Watson playing with these little cardboard cutouts of the bases. And with Donahue's correction, he suddenly realizes that the AT pair and the CG pair are structurally almost identical. They're the same length. They fit perfectly inside the helix, held together by hydrogen bonds.
It's that aha moment, a moment of profound, simple recognition after years of intense work.
And that solved it completely. The anti-parallel double helix immediately suggested how it could replicate. The 2 strands just unzip, and each one acts as the template to build a new partner. It was elegant, it was explanatory, it was revolutionary.
The discovery was made February 28, 1953, published in Nature a couple months later, and then Crick, Watson, and Wilkins get the Nobel Prize in 1962. It didn't just explain what genes looked like, it explained how they work.
It was the molecular language of life. And while the sources make it clear that Franklin's brilliant work was the key evidence, the double helix became the undisputed icon of 20th century science.
So with the structure solved, Crick doesn't rest for a second. He immediately pivots to the next massive problem.
He does.
Okay. We have the structure, the beautiful container. Now, what does it say? How does that sequence of bases actually tell the cell how to build a protein? He needed to crack the code.
And this is the period that really establishes Crick as this towering figure in theoretical molecular biology. The problem of linking the nucleic acid language to the amino acid language of proteins was the next great barrier.
So what was the big intellectual hurdle? They knew the structure, but the amino acids didn't seem to have any chemical attraction to the DNA itself.
That was the exact problem, and he solved it with pure logic, with theoretical necessity. In 1956, he proposed the existence of adapter molecules.
Adapters.
He reasoned that since there was no obvious chemical relationship, there had to be an intermediary, a go-between. These adapters, which we now know as transfer RNA or tRNA, would physically link to a specific amino acid on one end, and on the other end, they would hydrogen bond to a short complementary sequence of RNA. This was a purely theoretical deduction. He was describing a molecule that had to exist for the system to work before anyone had even found it.
That's incredible foresight, just defining a molecule based on its function.
And at the same time, he was wrestling with the math of the code itself. There are only 4 nucleotide letters, A, T, C, and G. So if the code used pairs of letters, you'd only get 4 times 4, or 16 combinations.
Not enough for the 20 common amino acids.
Not enough. So logically, mathematically, it had to be a triplet code, 3 letters.
Which gives you 4 times 4 times 4, or 64 possible combinations.
Exactly. And Crick realized this meant the code had to be degenerate. Multiple 3-letter words could specify the same amino acid.
This intense focus on information flow is what leads him to introduce one of the most famous and sometimes misunderstood terms in biology.
The central dogma.
The central dogma, which he coins in 1958.
And its purpose was to summarize the unidirectional flow of genetic information. DNA makes RNA, and RNA makes protein.
DNA, RNA, protein.
But his main goal was to draw a boundary. He was saying information can flow from nucleic acid to nucleic acid and from nucleic acid to protein, but, and this was the crucial part, information cannot flow backwards from a protein to the nucleic acids. That step, he called it irreversible.
But the word dogma is so interesting. It sounds so authoritarian, like an unquestionable rule, which seems completely at odds with Crick's whole skeptical personality.
It does, and he actually expressed some regret about that word choice later. He said he was using it in an older sense, meaning a compelling idea put forward without, rock-solid proof just yet. He was trying to provide a powerful hypothesis, a directional arrow for all the research that would follow.
So let's use an analogy for the listener. If DNA is the master blueprint in library archives, where do RNA and protein fit in?
That's a great way to think of it. The DNA is the master copy that can never leave the vault. So to build something, you have to make a temporary working copy. That's the messenger RNA, the mRNA.
Okay, the photocopy.
The photocopy. That mRNA then leaves the nucleus and goes to the construction site, the ribosome. The ribosome reads the code, and the tRNA Crick's adapter molecule acts like a little delivery truck. It brings the specific amino acid, the building material, that matches each 3-letter word, and the result is the assembled protein, the final machine.
So that 1958 paper was basically the entire framework. He laid out everything molecular biology would spend the next few decades proving.
He did. He outlined all 4 necessary components. 1, DNA stores the info. 2, mRNA carries the instructions. 3, the tRNA adapters mash the code to the amino acids. And 4, the ribosomes act as the factory to build the protein.
But it was still a theory until they could prove the code was, in fact, read in triplets. What was the experiment that proved that?
That was one of the most elegant genetic experiments of the era. It was work done by Crick, Sidney Brenner, and their colleagues in 1961. They used a virus, a bacteriophage, to do what are called frame shift experiments. Yeah, they used chemicals to either insert or delete single bases, single letters in a viral gene. When they inserted one letter, it shifted the entire reading frame of the gene.
Threw the whole thing off.
Exactly. The rest of the message became nonsense, and the virus couldn't make a functional protein. Same thing if they inserted a second letter. Still nonsense.
But here's the brilliant part. When they inserted or deleted a total of 3 letters,
the reading frame shifted, but then it shifted back into place. They had essentially added or removed one complete 3-letter word. The resulting protein might have 1 extra amino acid or be missing one, but the rest of the sequence downstream was read correctly again.
And that proved it. The code is read in units of 3. It's just a stunning piece of intellectual work.
It was the empirical proof for the whole theoretical framework. Later on, Marshall Nirenberg would work out the actual dictionary, which triplet codes for which amino acid, but Crick and Brenner gave us the grammar, the rule book.
And during this period, you get a really vivid picture of Crick's personality. He was known for being incredibly talkative, almost booming.
And that infectious, reverberating laugh, his presence just filled a room. He was described as a brainstorming intellectual powerhouse.
How did that intensity come across in the lab? Was he difficult to work with?
One colleague said he was never mean-spirited, just incisive. He had this amazing ability to spot microscopic flaws in logic almost instantly. He'd challenge an idea before the speaker had even finished saying it.
That intellectual rigor probably explains James Watson's famous quote that he had never seen Francis Crick in a modest mood.
He had immense confidence in his analytical abilities, and the thing is, he was usually right.
And this combination of fierce precision and this big, often irreverent personality, it's really important context for the next part of his life, when he starts wading into philosophy and some pretty serious controversies.
As we move past his core achievements in molecular biology, you really see Crick applying that same fierce, skeptical intellect to these much broader philosophical and even cosmic questions.
Let's touch on his legacy first, just in terms of honors. He got a ton of awards, the Order of Merit, but he famously refused a knighthood.
He refused the CBE in 1963 and later the knighthood. He just seemed completely uninterested in that kind of social status. He kept his focus on the science.
His Nobel medal was sold at auction.
Right. Yeah, in 2013 for over $2 million. And a big chunk of that, 20%, was donated to the Francis Crick Institute in London, which is this massive, modern biomedical research center. It's really a powerful physical testament to his vision for science.
Okay, now on to the deeper controversies. The sources note that he occasionally voiced some views on eugenics.
Right, and here it's important that we're just impartially reporting what the source material states.
Of course.
In private letters, he advocated for a form of what's called positive eugenics. He's quoted as believing it was unavoidable that society would eventually start to worry about the character of the next generation.
So what was he proposing?
His suggestion was to encourage wealthy parents to have more children. It seems to have stemmed from this sort of intellectual meritocratic viewpoint, but he was also cautious, saying society wasn't ready or ethically uniform enough to attempt something like that.
And that kind of thinking, combined with his rigid atheism, it places him firmly in the humanist camp.
Very firmly. He defined humanism as the belief that you have to solve human problems with human resources. You have to rely on rational inquiry and evidence without invoking supernatural authority.
He was known for being particularly harsh about religion.
Oh, very much so. He called Christian beliefs ridiculous. There's a famous joke where he said Christianity may be okay between consenting adults in private, but should not be taught to young children.
He had zero tolerance for ideas he saw as irrational.
Zero. And it played out in his professional life, too. He actually resigned his honorary fellowship at Churchill College, Cambridge.
Why?
Because the college accepted a large donation to build a chapel. He saw the mixing of science and religion as just fundamentally incompatible. It shows how deeply he held that secular worldview.
And this philosophical conviction that everything can be explained by physical laws, that's the foundation for his later work on the brain, which he laid out in his book, The Astonishing Hypothesis.
Exactly. In that book, he states his view so clearly. The idea of a non-material soul, he says, is just an imagined idea, a metaphor. The mind is purely 100% the product of physical brain activity, which itself evolved through natural selection.
He even predicted that once we fully understood the brain's detailed workings, what he called erroneous Christian concepts of the soul would just be replaced by scientific explanation.
He even suggested testable ways to bridge that gap. Back in 1970, he speculated about something he called biochemical theology.
Biochemical theology.
Yeah, he suggested that if prayer is a real phenomenon, it must have a measurable physical effect. He speculated that you could probably measure chemical changes in the brain neurotransmitters, hormones that correlate with the act of praying.
Which is basically the field of neurotheology today. He was way ahead of his time on that.
Absolutely. And his curiosity didn't stop at the brain. It went all the way to the origin of life itself. He knew so much about molecular complexity that he found it really hard to believe that life could have just arisen spontaneously from chemicals here on Earth.
So that led him to a pretty wild idea.
It led him and Leslie Orgel to co-develop the concept of directed panspermia in the early 70s.
Directed panspermia. What is that?
Well, they were just so puzzled by the complexity required for that first living cell to assemble itself. They argued that maybe life is such a rare event in the universe that it was just too improbable to have happened randomly on Earth in the time available.
So instead of a random terrestrial origin, they proposed
What? They speculated that maybe life started somewhere else, on a much older, more stable planet, and then it was spread to Earth intentionally by an intelligent alien civilization using some kind of spacecraft.
Wow.
It wasn't really a scientific theory, it was more of a philosophical thought experiment. It was their way of saying, look, the problem of the origin of life is so hard, we shouldn't rule anything out.
It says so much about his character that even after defining the architecture of life, he was willing to entertain these incredibly radical ideas.
He was a radical thinker. He reserved the right to question any assumption, no matter how fundamental it seemed.
So then, in 1977, he makes what has to be one of the most radical career moves in the history of science. He's 61 years old. He has the Nobel. He's the undisputed king of molecular biology. And he leaves Cambridge.
He leaves Cambridge to join the Salk Institute in La Jolla, California, and he dedicates the entire final phase of his career to theoretical neurobiology.
I just can't get over that. He's at the absolute peak of his field.
The field he helped create.
Right. And he just walks away. Start over as, like, a student again in a whole new field.
At age 61. It's an incredible move. And you have to remember, molecular biology was still on fire. I mean, they were discovering restriction enzymes, gene splicing.
All the stuff he helped make possible.
And he just turned his back on it to go chase the biggest ghost of all, consciousness.
So when he gets to neuroscience, what does he find?
He finds a field that, compared to the beautiful logical system of molecular biology, was fragmented and, kind of primitive. And he immediately identifies three big challenges that were holding it back.
Okay, what were they?
First was just the sheer fragmentation. You had all these isolated subdisciplines, anatomy, physiology, psychology, and they barely talked to each other. There was no shared language.
I see.
Second was what he called the black box approach. A lot of researchers were just studying behavior, the output, without really trying to look inside the brain to see the mechanism that was producing it.
And the third problem was the biggest one of all, the taboo.
The taboo around consciousness. It was considered too difficult, too philosophical, just not a subject for serious empirical science. And Crick was determined to change that.
So how did he attack this massive, complex problem? Where do you even begin with consciousness?
His approach, especially in his long collaboration with Christoph Koch, was to narrow the focus dramatically. They knew they couldn't solve all of consciousness.
Right.
So instead, they decided to focus on a measurable phenomenon, the neural correlates of consciousness, specifically how the brain generates visual awareness in the first few hundred milliseconds after you see something.
Why visual awareness?
Because it's fast, it's repeatable in a lab, and it lets you draw a clear line between what the brain processes unconsciously and what actually breaks through into your conscious perception. They wanted to isolate the physical mechanism that made that difference.
And did they have a hypothesis for what that mechanism might be?
One of their big ideas involved neuronal synchronization, particularly something called 40 hertz gamma oscillations.
Okay, what's that?
It's when different groups of neurons across the brain all fire together in a synchronized rhythm about 40 times per second. Crick and Koch hypothesized that this rapid synchronized firing might be how the brain binds different features of an object, its color, its shape, its motion, into a single unified conscious perception. It's the mechanism that tells your brain, hey, these features all belong to the same thing.
So in the astonishing hypothesis, he's really arguing that the field is finally mature enough to tackle this stuff scientifically.
Yes. And he also hypothesized that consciousness seems so mysterious to us because it involves these very short-term memory processes that we don't understand well. If information isn't held in that immediate, accessible buffer, we don't feel conscious of it.
His work wasn't just about awareness, though. He had this fascinating idea about dreaming.
The reverse learning hypothesis, which he developed with Graham Mitchison in 1983.
Reverse learning.
The theory is that the function of REM sleep, of dreaming, is basically unlearning or neural housekeeping. Your brain is constantly forming new connections, and some of them are just junk, parasitic, noisy connections that aren't useful.
So dreaming is the brain's way of cleaning up the junk files, deleting the clutter.
That's a perfect way to put it. The reverse learning process weakens or deletes those unwanted connections to keep the whole network stable and efficient. It's a computational theory of sleep.
It always comes back to information management for him, doesn't it? Whether it's genetic information in a DNA sequence or network information in the brain.
He was a scientist until the bitter end. The sources say he was still at the Salk Institute, working until his death in 2004. He was actually editing a manuscript on his deathbed. He just never stopped chasing the physical answers to life's biggest questions.
So when you look back on his whole career, the sheer breadth of it is just, it's astonishing.
It really is. He gave us the structure of DNA. He gave us the central dogma, which completely changed how we see life as an information system run by a code. And then he successfully pioneered the scientific study of the most complex thing we know of, the conscious mind.
He fundamentally shifted the scale of our understanding. He took biology from a descriptive science to a science of mechanism and information.
Yeah.
And that legacy is so physical, too. The Francis Crick Institute in London is this massive tribute to the kind of rigorous, integrated science he championed.
He taught us that the deepest mysteries of biology can be and must be solved with the tools of physics and chemistry. He took us past the question of what life is made of to the question of how life works.
Which leads us to a final, provocative thought, building on that relentless pursuit. Crick was totally convinced that by understanding the physical brain, we would eventually just do away with our ancient concept of the soul.
Mm-hmm.
But in his own field, molecular biology, we've now gone even further than he could have imagined. Synthetic biology is creating brand new synthetic DNA molecules that use more than the standard 4 bases. They're expanding the genetic alphabet.
Yeah.
So if we can now not just read life's code, but actually rewrite it, and maybe one day follow Crick's ultimate dream of synthesizing life from scratch in a test tube using this expanded synthetic DNA, does that finally put to rest the distinction between the living and non-living that he sought to bridge? Once we can write the code and invent new life ourselves, does the mystery of life's origin stop being a cosmic speculation and just become an engineering problem?
Yeah.
That is something worth pondering.